Man, what happened here? This book has so many mistakes. It's never gonna make it. People can be like this too. Like, that kind of sounds rude, but human genetics can be pretty whack. There's sickle cell, HIV, cystic fibrosis, and all sorts of cancer. Well, now I'm kind of sad. Can these be treated?
We have drugs, but they don't always work. There's gene therapy, but pretty much only the Prince of Monaco can actually afford that.
So what's the hope left in this Pandora's box of disease? CRISPR. So CRISPR is part of a bacterial immune response that could cut up viral DNA. It can be applied to edit genomes at a specific locus.
There are three main components of the CRISPR-Cas9 system. First, there's the Cas9 nuclease, which is an enzyme that cuts through both strands of the genomic DNA. Then we have the tracer RNA, which activates or turns on the Cas9 nuclease and connects it to the third component, CRISPR RNA, which recognizes specific sites on genomic DNA and guides the rest of the complex to the target sequence. This CRRNA sequence can be programmed in the lab to provide the versatility of CRISPR-Cas9.
Think of the CRISPR-Cas9 system like some word processing functions. Here, as I edit this error-ridden book from the beginning, I could search for a specific part I want to change using the Control-F function, or our CRRNA. I found my target and will now make a double-stranded break with the Cas9 enzyme. This autocorrects into a mutation that can allow scientists to determine the function of a gene. But when we want to edit a sequence, we could cut out the mutation and add in a new segment. One limitation is that if we have different organisms, we can't necessarily use the same CRRNA to treat the same disease. Just as deleting one word can't always fix mistakes in two different books. You may have heard about a recent study where researchers were able to edit a human embryo using CRISPR-Cas9. They removed a mutation that causes the thickening of cardiac muscle, a condition called hypertrophic cardiomyopathy. The amazing thing about this study is that researchers were able to edit the mutated gene without any unwanted off-target modifications. Furthermore, they were able to edit all of the cells in the embryo so that they all had the same gene sequence. CRISPR-Cas9 has so many applications in the treatment of genetic abnormalities, such as HIV, sickle cell, and cancer. Maybe someday we'll be able to detect and treat genetic diseases even before we experience their symptoms. So maybe this book does have a chance to make it big someday. Thanks, CRISPR.